like. Anyway, it's with mixed emotions, but mostly sadness that I introduced today Etienne, whom you all know, and who's going to give the last talk as an IMO member before he moves on to greener pastures. Etienne moved here to Tampa about 5 years ago to work with us and with our experimental collaborators, Connor Lynch, and with later on Andriy Marusik. He has managed to get great research, and he has managed to make lots of good friends, which I hope that you keep up with as you move to Canada. I know that you're going to tell us a tiny little bit about what kind of work you'll be doing in the future. I know it's not going to be math onco, but I know that a lot of the talk today is going to be covering some of the projects you've been working in the last few years, who have led to some very interesting publications. So, Etienne, please, the stage is yours.
Thank you, David, for the kind introduction, and thanks everyone to be here. So I'm going to go through mostly two main projects that I've been working here in Moffitt. And so the first one stemmed from the goal to understand better biology of bone metastasis, as it's a big deal clinically. 90% of patients that die from prostate cancer exhibit bone lesions, and 80% of the prostate cancer that metastasize will spread into the bones. And as you can see, the survival for late stage disease is very poor. And mainly prostate to bone metastasis remains incurable and consists in painful osteogenic lesions, and massive, bone remodeling and heterogeneous, like bone, density and quality. And one thing that is critical in the progression of bone metastasis is the crosstalk between prostate cancer cells and the bone microenvironment. And maybe the way it's playing out, at least according to most of the studies, is that prostate cancers will stimulate precursor osteoblasts and osteoblastic cells that will in turn activate osteoclasts to resorb bones. And from this resorption, a lot of growth factors cancer cells can take advantage of will, drive further cancer progression into the bone. And this is already a very complex dynamical system that is, fairly poorly understood so far.
Etienne, we can't hear you. I don't know what you did there. Oh, we can hear you now. I don't know if you bashed your mic or something.
Oh, okay. I'm sorry. So can you hear me now? Is it okay?
Yep.
Yes.
Okay.
Yes.
So we missed this slide.
Okay. So basically here, our goal is to understand better the underlying biology in combining in vivo mouse models with in silico models, and to basically compare some predictions to observations. But one main issue with that is even the bone biology itself, so without presence of any cancer cell, is not very well known in terms of dynamics and what are the cellular actors that play the different important part of bone dynamics. And in order to understand the system, we develop a in vivo bone injury model where an intratibial injection is performed, and then we monitored the main bone cell population as well as the bone dynamics. And so typically over few time points, osteoclast numbers are tracked with TRAPC marker. Osteoblast counts are performed using the RONX2 marker, and bone volume is tracked using micro-CT analysis. So from these data, we performed 2 studies, basically aiming to understand 2 different aspects of this bone injury repair process. The first one is really about bone formation and resorption rates quantification. And the second is more about the bone cell population dynamics and in a sort of couple system. So for the first study, basically, so as I mentioned just previously, this is a schematic of our bone injury model with basically the experimental data. And from the experimental data mentioned, we can obtain some temporal plots of, so in black, the bone mass, in gray, the osteoblast counts, and in orange, the osteoclast counts. And for those that are not super familiar with bone biology, osteoblasts are the cells that form new bone matrix, and osteoclasts are the cells that resorb bone matrix. So typically when bone is damaged and you need to regenerate your bone, osteoclast will resorb the damaged bone and osteoblasts will form some new fresh bone. And so first we aim to model the system with a very simple math model with mainly 3 variables, osteoblast, osteoclast, and bone, which are the 3 equations. And the 4th equation is basically imposing that the bone resorption term always equate the bone formation term when all the species are at homeostasis. And so I would say the bone equation is the most simple one. And it contains a bone resorption term that is proportional to osteoclast and a bone apposition term that is proportional to the number of osteoblasts. And basically the osteoblast and osteoclast dynamics are described with a very phenomenological model here, where we just consider homeostatic terms and intermittent expansion for osteoblasts and intermittent decrease for osteoclasts. And the goal here is not really to have a very mechanistic model of those species, but more to understand whether when we have a function that fits well to those dynamics, can we predict accurately bone dynamics? So we perform this fit and then try to predict the bone. And in order to predict the bone dynamics, we basically retrieve parameter from the literature. And in this study, osteoclasts were cultured on dentine slice, and they form pit in the slice. And so the researchers, measured at what rate the osteoclast resorb bone. So in the last column, estimated volume of bone that is resorbed per unit of time. And you can see amongst the 6 osteoclasts that are here that there is already a really big variability, inter-cell, because we go from 43 micrometer cube per hour to 1,000, more than 1,000. And the answer is that we can't predict accurately the bone mass with those rates. basically we overestimate the bone mass, and it means that, you know, fixing the resorption rate to those in vitro rates and imposing some homeostatic condition, that is the fourth equation, we can't recapitulate the data. And one main assumption that is made here is that the bone resorption rate and apposition rate per cell is constant over time. And we'll see that this assumption is probably very arguable. And so, you know, so that's the same model, but the dash part is the, you know, prolonged like prediction to longer term. And the D, when you look to the D plot, now it's not predictions anymore. It's so we still maintain the two osteoblast and osteoclast fits, but we allow the bone resorption and apposition rate to be freely fitted to the bone dynamics, but without imposing the homeostatic condition anymore. And so when we do that, we obtain a good fit, as you can see, but the predicted rate, the predicted bone mass in long term will be much lower than the actual bone, you know, homeostatic level that was measured. And so it means that it's hard to, you know, reconcile these bone dynamics with the assumption that the bone resorption rates and apposition rate per cell are constant, because in that fit, that's still the assumption. It's just that now we're free to calibrate those rates, but still we can't, you know, this time we can recapitulate the observed part, but the predicted new homeostasis is half what it should be. And so so these are the, you know, estimated parameters with the uncertainties that go with the estimation. But how to explain these paradoxical observations? Well, a hypothesis would be, and that's our main hypothesis here, is that osteoclast and osteoblast activities vary over time. And this variation over time could potentially be mediated by some other cells that are present in the environment. For instance, yeah, this publication shown that osteoclast rates are increased by a good margin in presence of some pro-inflammatory cytokines like interleukin-1. And in the context of bone injury, those pro-inflammatory cytokines are present in very high level, especially at the beginning of the process. And so it would make sense that you know, the cellular activity, for instance, osteoclast, would meet a spike during the process and then cool down to you know, more homeostatic activity. You know, a natural candidate cellular candidate in terms of pro-inflammatory cytokine is macrophages and monocytes that are present in high frequency in the bone, until 10%. So that could be, you know, a sort of a extended model of this one, which is osteoclast activity would be enhanced by pro-inflammatory macrophage, called sometimes M1, and osteoblast activity would be enhanced by anti-inflammatory macrophages that are called sometimes M2. And so to understand, you know, the role of these cells better, on the same like injury model in the, you know, contralateral tibia this time, flow cytometry was performed using immunomarker to characterize total monocyte and macrophage and the pro-inflammatory and anti-inflammatory subsets. And so now, if we want to, obtain a model that would recapitulate both bone dynamics that is observed during the process and that maintain the right homeostasis at the end, we try to basically extend the previous model in considering this time time variation in the bone resorption rates. And here, the choice of the functional form is a piecewise linear function, which are allowed to basically vary in terms of slope between each time point. So as you can see in the bottom orange curve, so different slopes were basically estimated between each time point, and you can see that with this model, which is very again phenomenological, not very mechanistic, we can obtain a good fit to the bone. And this time, because the piecewise linear function is imposed to come back to the homeostatic rates of bone resorption, it will be able, the bone dynamics will accurately fit the injury data, but as well come back to the right homeostasis. But what's more interesting in those plots is when you look to the temporal profile of the estimated function, so the orange function, which represents the predicted resorptive activity per osteoclast. So you see that sort of double spike temporal profile with one short high spike and then a longer lower spike. That temporal profile looks very similar to the temporal profile of pro-inflammatory cells that we have measured. So the green is the pro-inflammatory monocytes and the purple is the pro-inflammatory macrophage, so M1s. And so it makes sense with what we previously discussed about the potential role of these cells in osteoclast activity. And that kind of temporal profile inflammatory pattern has been observed in a broad range of inflammatory processes, not only in the bone. When we try to do the same thing, so the piecewise linear function, but not for resorption for a position, we don't manage to get as good as a fit. And then what we did next is now we include some pro-inflammatory macrophage and monocyte and anti-inflammatory macrophage dynamics, and we link the bone resorption rate and apposition rate proportionally to the level of those cells. And we can obtain a good fit. And because these cells come back to a homeostasis that is close to 0 at the end, we can recapitulate the right bone dynamics and come back to the right homeostatic bone level. And doing so, we estimated some variation in osteoblast and osteoclast activity. And when you look to the two last rows of the table, which are the bone formation range that is estimated per cell and the bone resorption range that is estimated per cell, you see that we have a dramatic variation for the bone resorptions as expected, but not as big as a variation for the bone formation rates, just a factor 2, whereas it's more than an order of magnitude for the bone resorption. So what did we learn about this study? Well, osteoblasts and osteoclast numbers are not a good quantitative predictor of bone dynamics. And bone resorption and apposition rates are altered during the course of injury. Bone resorption optimal functional shape follows inflammatory dynamical pattern. So our hypothesis that seems supported by our data so far is that polarized macrophage have a role in not only potentially the population dynamics of those cells, but as well their activity. And that's the goal of our other study, which is trying to recapitulate the bone population dynamics. And again, especially the pro-inflammatory macrophage population has this complex sort of two-wave profile in our data, and we're interested in trying to understand what kind of mechanism could recapitulate overall this pattern, because it's not very well understood. And so in order to do that, and because we have so many cell types to consider here, so we have like 8 observables, which are osteoblast, osteoclast, pro-inflammatory monocyte and macrophage, anti-inflammatory macrophage, bone dynamics, and total as well, monocyte and macrophage. This dynamical system is very complicated and coupled amongst these species, and so several part of those dynamics are just partially understood, meaning that multiple mechanisms have been shown to drive, like, let's say, the expansion or the decline of these different populations. And so what we try to do here is to build a model that recapitulates all the cell population dynamics based on published mechanisms. But what we try to do is for each cell population, try to not be redundant in accumulate different mechanisms, even if they have been shown to play out. And so, for instance, for osteoclast dynamics, it has been shown, at least in vitro, that M1 and M2 have a positive and negative impact respectively on the cell population, but so did osteoblast, for instance. And for osteoblast, it's even more complicated because osteoblastic cells produce both pro or anti-osteoclastic factors, depending on the context. And so, for each, like, basically point that was interesting to cover in terms of cell dynamics, we propose a list of different, like, candidate mechanisms and combining. So let's say I do A1, B1, C1 would be one model. And so we tried all the different combination of assumptions and compared the goodness of fits against the data. And so the most interesting one here, piece of dynamic, is the third one, which is the macrophage dynamics. So the first mechanism is that injury factors basically drive both M1 and M2 at the same time, with M2s producing, an anti-inflammatory array of cytokines that will act as a negative feedback on the pro-inflammatory cells. The second one is that in order to elicit an anti-inflammatory reaction, you need inflammation first. And so the pro-inflammatory cell will basically elicit that anti-inflammatory reaction, and there will be a negative feedback in return. And the third one is basically about plasticity, which has been suggested by a certain number of studies as well. And it's that basically, initially on the injury sites, pro-inflammatory macrophages will be recruited, and when they basically uptake the cellular debris by phagocytosis, they will reprogram into an M2 phenotype, and so that could be as well what plays out here. So amongst these 18 different combinations, that's the one combination that produced the best fit. So A3, B2, C2, and basically in terms of macrophages, that's the second mechanism that was retained, which is pro-inflammatory cells eliciting an anti-inflammatory reaction. And so you can see that we get a very decent fit for all the populations. And in order to rank the different models, we use the ASE criteria, although the number of parameters is almost the same for all the models, but there are some variations here and there. So that's why we consider the ASE. And this is the equations. So we have the 8 variables, which has the 8 first equations, the 8 variables that are observed, and the last equation is represent the amount of cellular debris present in the injury, and it's getting cleared over time by pro-inflammatory macrophages. And that's an example of one model that fits poorly the data. So you can, just for the sake of comparison, I'm not going to show the 18 models because I don't want to give you guys a headache. But yeah, that's one example, and you can see that some of the populations really, like the model really doesn't capture what's happening. So in that study, basically, what comes out of here is that osteoclasts are massively inhibited early in the process, and our modeling approach suggests that the main contributor, and really we had like 2 candidates for that, osteoblasts and macrophage and M2 macrophages, and so suggests that the main inhibitor source is M2 macrophages, and the 2 waves pattern for the M1 macrophages was challenging to recapitulate because most of the model couldn't. But the one mechanism that could recapitulate that was the M2 mediated inhibition of the M1 macrophages, as opposed to the plasticity hypothesis that fails to recapitulate our data. And so, yeah, so these studies, so one has been accepted in a scientific report, and the other one is under review right now. But based on those results, basically, the next step was to try to incorporate cancer in that system. But this system is already very complicated, and so adding cancer would, make the model probably way too complicated to understand and handle. And so, typically, cancer cells have been shown to be able to educate macrophage, in a lot of tumor tissues. A spectrum of macrophage phenotype have been observed. Some tumors are highly infiltrated by M2s, some by M1 and sometimes by both. But probably when it's both, it's a distinct spatial niches. And that's what we're trying to investigate here. And in order to investigate that aspect, we consider a bone metastasis agent-based model that comprise only tumor and macrophage. So we don't model explicitly osteoblasts and osteoclasts here. What we consider though is that M1 macrophage will drive bone resorption, and M2 macrophages will drive bone formation. And obviously not directly, indirectly by the enhanced osteoblast activity or osteoclast activity. Basically macrophages infiltrate by the vasculature in this model. And we will initiate the model with the presence of two phenotypes of cancer, which are stimulating M1 polarization or M2 polarization, respectively. And the white part in the medullary canal is the bone. So the bone overall density was based on the bone homeostasis that was measured by micro-CT. The microvasculature was taken, density was taken from literature, and the number of macrophage from the flow cytometry data. And so in that simulation, for instance, and for the color code, the orange macrophage are M1 and the blue macrophage are M2. So here, the purple and green cancer cells start to expand and polarize surrounding macrophages. And you have some part of bone formation that are, form this block of bones, but that are as well resolved at the same time. So M1 macrophage resolved in, M2 macrophage basically make them expand. And in this model, basically the basal fitness of cancer is fairly low, as well as the turnover. But when bone is resorbed, it releases some factors that will give them a substantial fitness advantage. So to obviously the cells that are in contact with the factors. And so ultimately in that simulation, we obtained basically sort of two cluster of cells. So the green that are osteogenic and the purple that are osteolytic. And in that second simulation, it's similar, but interestingly, the osteogenic phenotype surround the osteolytic phenotype. And usually very sort of a pattern that was very repeatable across the different simulation is that usually the two clusters of tumor phenotypes are separated by a big chunk of bone. And what's happening is that there is a front of resorption that will help tumor cells to proliferate, but the green cell will maintain the bone mass and so maintain sort of a substrate for bone turnover. And so it's really a sort of cooperative, I would say, phenotype, in the sense that if you have one phenotype or the other, the tumor won't expand as fast as if the two phenotypes are present. So here is another simulation, again, like chunk of bone being at the interface of the two, and same here. And in that one, though, the osteolytic phenotype is very dominant in that one. but this is the agent-based model is very work in progress. So those are really more like fresh results than the bone study. But the advantage of the bone study was that we could estimate parameters, typically bone resorption and apposition or polarization rates of macrophage that we are now using for the agent-based model. And we're aware that those rates could be different from the bone injury because it's a different context, it's cancer, but it gives at least some realistic parameter values to work with. And so what we observed so far in this spatial model is that we usually observe two distinct spatial niches, one osteogenic and one osteolytic. The coexistence of these two phenotypes allows faster colonization of the medullary canal than each phenotype alone. And across the simulation, we observe usually net bone mass, so an overall osteogenic cancer. And for future work for this, we'll require a more systematic quantitative analysis. So typically the temporal total quantification of each cell type and the bone mass, and maybe compare that to the flow cytometry data in like prostate cancer mouse models. And another thing that we are very interested in is comparing those spatial distribution of osteogenic and osteolytic phenotypes to actual stain of bone section of bone metastasis in mice, as some markers for that characterize well M1 and M2 macrophage can be used for those. And luckily, Connor, likes this spatial model and is willing to provide some of these kinds of data. So I'm actually going to talk about another project right now. I don't know what. Okay, still have some time, but I don't know. Maybe that's a good time for questions about this one.
Yeah, I think that is a good time. So if there's any questions on the kind of macrophage story, I definitely have one. So I'm kind of intrigued about the plasticity aspect there, and clearly it's not M1 or M2, it's M1 and a spectrum of states in between, and then
Right.
M2, right? So how important do you think that intermediate states could be?
That's a very good point, Sandy. And I'm not gonna lie, it could be very important. And yeah, thanks for bringing this up because that's actually an important point. So in our modeling perspective is what that one clear reason why we consider just two separate phenotypes just is way simpler to model. And a lot of biologists, but I think mostly non-immunologists, still consider that description to be useful and relevant in some contexts of just two discrete phenotypes. But more recent studies have shown that, first is not just two phenotype, but like you could distinguish between, for instance, anti-inflammatory and tissue regeneration. But it goes beyond that. It's probably more a continuous sort of spectrum of phenotypes. But that could be something to consider in the future. And I think experimentally as well, it's a challenge to quantify sort of a continuous distribution of macrophage, let's say. Because in flow cytometry, you have kind of a distribution. You know, if you look to, like I think I plotted it, but not sure. Yeah. Oh, never mind. The NAS2 and arginase-1 are not in that one. But you know, if you plot the two axes that characterize sort of M1 and M2, you know, as traits. So in our studies, NAS2 and arginase-1, the markers, you see sort of a continuous distribution in this 2D space, but mostly you see two clusters of cells, which we consider M1 and M2. Again, as well, the plasticity, what I'm saying about the plasticity model that doesn't really fit the data as well pertains to the bone. And I'm not claiming that it's valid for other tissues, because that could be different too.
Sure. No, it was a tough question. And I think my point was, like, it makes sense to simplify and model the binary states, and that's the way the world thinks. But there is a kind of timescale issue in some sense about switching between those states that doesn't really exist if there's just two states, right? Because you can just switch, versus if there's now intermediate states where you've got to transition through those, and that could take some time, right? And so there, you know, there could be a sort of time scale associated with that, which could be interesting.
Yeah, absolutely.
But we don't need to discuss it any longer. But,
but I think recent sort of single cell data suggests that it's not like discrete states. It's really kind of a continuum of phenotype that the cells go through.
Yes, that's right.
But I have a quick question. So what is the genesis, so to say, of the M1, M2s? So what is their directly above lying cell type in the differentiation hierarchy, if you will, and
so I'm not sure I understand fully.
A common progenitor
Philip,
or something.
Right. So in terms of lineage, you mean, is there a link with the maturity of the cells in the? I think, you know, it's not like entirely uncorrelated. I think there is a link. Like, for instance, less mature macrophages that are more in a monocytic state. They're more
exactly, monocytes, right? I forgot about that. Yeah, okay.
Yeah, they're more prone to turn into a pro-inflammatory phenotype. Whereas when they mature, for instance, when they homed into the tissue for a certain time, they're more prone to polarize into an anti-inflammatory state. And so that's why that, you know, most of the NOS2 positive cells that we see are monocytes, and most of the arginase-1 positive cells that we see are, you know, macrophage or more mature. And by mature, I mean they lose the Ly6C marker expression.
Okay, yeah. And so if you want to go down the road of cooperation to explain coexistence, you have to somehow be able to show that there's a cost involved with one or the other and a benefit, and how this is context dependent, so that they, I mean, there can other, right? I think it's very interesting to maybe explore different ways of coexistence that are not exclusively cooperative. There are others.
Yeah, and I'm that's why I use the question mark as well, is like, at this stage, I don't know if we can talk about cooperation, but it's more, I guess, the osteogenic cells benefit from, you know, the osteolytic cell by, you know, bone turnover, and they maintain that bone mass. But yeah, it's at this stage it's unclear to me if there is a cost, maybe in the a cost to polarize macrophage into you know, the more osteogenic or osteolytic phenotype. But I haven't really considered any cost at the moment. Would that be interesting?
One thing I was wondering, yeah, I guess, is related to the cost issue. Is I guess this is sort of related to the progenitor question. Is that the progenitor step before you get to macrophages is sort of, I think it's probably like the last point of like exponential amplification in the hematopoietic differentiation process. So, and in a lot of other models, I've seen that basically when you have differentiation into one lineage, it necessarily means a trade-off into differentiation from the other, right? So I'm wondering whether the other kind of like granulocyte dynamics that are being impacted, or whether you have the data, would be useful.
For granulocytes, you said?
Yeah, basically that, if a progenitor is differentiating into a macrophage, right, it's at the very least time that's not being spent differentiating into another type of cell. And
right.
On a longer time scale, it's probably just, mutational cost or finite or near or finite, but still very large proliferative capacity that's being used, depending on where on that debate you side. But yeah, I think that, and I just wonder, just because there is that exponential amplification, right? So you might think that like a modest increase in macrophage cells might not have a significant cost, but if that modest increase necessitates a shift in a progenitor that would then lead to an exponential deficit, right, in another type of lineage, it might be interesting to look at. Although I don't know the bone ecosystem enough to know whether or not the other lineages would be significant problems for the remodeling.
Yeah, I think that'd be very interesting if we had a clear like progenitor that we can characterize well, like by flow cytometry, for instance, that can commit either macrophage or granulocytes. I know our experimentalist collaborator initially aimed to quantify granulocytes as well, but I think that the markers it was hard to characterize, but that'd be interesting to see how, let's say, the pool of progenitors is able to maintain when you have a substantial commitment to one or the other lineage. And if we can recapitulate all that, I'd love that.
Yeah. I mean, I'm not sure how far back in that proliferation tree you need to go. But the other thing I was wondering was, do they give like any type of GCSF when they're undergoing treatment for these metastases, or is it a situation where, I mean, I know that some cancers, they do give it for chemo, right? So as just like almost prophylactically for chemo. So I'm
like
wondering,
in terms of when in case of neutropenia, or
yeah, basically in case of neutropenia, the other major time that it's used is when you need to do like a bone marrow transplant. So when you're collecting it, right, they just pump you full of GCSF because it also causes your stem cells to leave niches and go into circulation. But yeah, I mean, I'm just, because looking at it more like in the therapeutic context, right? Like I feel like there are a lot of also this artificial external levers that were being pulled on in the clinic to try to prevent some of this. Like, did you mention that they, I think, yeah, you mentioned that they sort of give hormones to, maybe I'm thinking of something else, for preventing bone loss?
So in the case of advanced prostate metastatic prostate cancer, they give, to prevent, and as a palliative treatment, they give a biphosphonate that limits bone remodeling, basically. But yeah, I'm not sure if that's what you referred to.
Yeah. Sorry, that was what I was wondering about, because I wasn't sure. Because it seems like the remodeling process, could get out of hand very quickly. So I'm wondering how the patients
I know for macrophage targeted therapy, not only for the bone, but at least preclinically, there have been studies that tried anti-MCSF, to prevent macrophage recruitment in the tumor tissue. So yeah, I mentioned that because you were mentioning like CSF, but they tried like those anti-MCSF to prevent macrophage maturation. But in the bone, I'm not sure if anyone tried this.
Yeah, I would. I mean, we probably have to dig into like maybe some clinical trial stuff and see what, because that would, I mean, that would be written in the protocol, but it probably wouldn't be highlighted. Okay, yeah, thanks. I was just kind of curious about that.
Yeah, thank you. So if there's no more question, I'll talk about the other project.
I guess I got one real quick. I'm curious about your spatial model. Like you said that Dr. Lynch was interested, and he's going to, hopefully his lab's going to produce some stains, tissue slides to kind of work with that model. But I'm curious if, just because you showed some histology before, if the model design here is at all inspired by the data you already have, or like how you might connect the model to the histology. Because I think that's sort of, in a lot of ways, the most challenging part of building spatial models, is how you connect that to the actual biology.
Yeah, I agree. Actually, here, I guess, yeah, there are two things I would like to do in terms of, make the model meet reality. The first one would be more macroscopic data that measure by flow cytometry, so the total macrophages, the amount of pro-inflammatory and anti-inflammatory macrophages and the tumor cells, so by flow cytometry typically, and by microsity the total bone mass. So if we can recapitulate that, but because it's spatial, that'd be probably even more interesting to compare that to actual spatial distribution. And the good thing about those phenotypes is that at least there are some markers that work well in staining, to characterize these two extreme macrophage like phenotype, even if it's more a continuous sort of spectrum. And so, for instance, are we observing in bone met those clusters of M1, M2, and is there like those bone interface? I'm very curious about that. But yeah, I haven't seen this kind of data yet. So I had seen similar data like with M1, M2 markers, but for normal bone, but not for bone metastasis yet.
Thanks.
So yeah, the other project is, so we're gonna switch a topic in the sense that in the other project we were more interested in bone and tumor ecology, whereas here I'm going to talk mostly about cancer evolution. And cancer evolution is usually thought as this branching evolution, so asexual process when diversification emerged from replication error after division. And not only in cancer but in nature in general, asexual evolution has several implications. And first one is clonality, where well-adapted individuals can clone their advantageous genes. So that's usually an advantage. But there are some drawbacks to asexual evolution as well, is that it provides a low amount of diversity compared to parasexual and sexual evolution. And so in the case of environment context change, that can be an issue, or lead to different dynamics like selection. And another point is that some good mutations get stuck with loser mutations and don't get a chance to be selected for. Whereas for, when you have like genetic recombination, eventually you can, with luck, conserve like most of the good mutations and leave loser mutation to one cell that will eventually be selected against. And as well, there's the issue that maybe I'll mention later in small populations, that accumulation of deleterious mutations can lead to extinction. And cancer is seen mostly as an asexual evolutionary process. But is that the case? Well, most of you know in more or less detail how cell division works. And maybe as well about cell fusion, because that's actually a physiological process that happens in some of normal cells, like fusion between monocytes to form osteoclasts in the bone, for instance, and some, muscle cells as well fuse together sometimes. But actually, that process happens in cancer too, and that makes sense that because it's a physiological normal process, cancer could eventually get access to it. And in this video, you'll see, green fluorescent cells and red tag tumor cells that are, in co-culture, and some will eventually fuse and form, yellow hybrid. Yeah, so the hybrid appeared. Yeah, so here the fusion occurred here, and you can see that then the yellow hybrid will divide and give rise to, several daughters and so on. And so, so this was, these co-cultures were performed by, Andre and Daria, and they actually developed this nice experimental pipeline to systematically quantify a certain number of hybrids in a co-culture. So basically, you have a cancer cell lines, and half is green tagged, half is red tagged, and after 3 days of culture, they quantify the double positive cells as hybrids. So at left, by flow cytometry, you have the total amount of double positive, but they use as well video microscopy to basically distinguish the eventual false positive amongst this double positive versus actual real fusion events. And so spontaneous tumor cell fusions were observed in vitro, but as well in vivo for a certain panel of cell lines, like MDA231 and some 159. And here you see the video microscopy that allows to distinguish, for instance, between actual cell fusion, entosis, which is when a cell engulfs another one, and some cells that just overlap. And the number of double positive events was quantified for all the cell lines they used. And you can see that in the most like fusogenic cases, after 3 days of co-culture, it reached a 0.6% of hybrids. So the question then is, so that those hybrids like emerge, but what are they doing? Like, are they able to divide? Are they able to pass on their phenotype? And in order to answer that question, Andre and Daria developed these co-culture associating green fluorescent protein expression with elasticity and resistance, and M-cherry expression with pure myosin resistance. And then by the challenge of double antibiotics, they will obtain the double resistant cells, which are hybrids that form single cell colonies. And so the clonogenicity of the hybrids was, quantified out of this. And they managed to obtain single cell colonies of hybrids, which shows that some of these hybrids are actually clonogenic. They can form a colony from just one cell. They measure as well the growth rates, the migration potential, and basically if anything, the hybrids were mostly more migratory than the parental cells. They had as well enhanced metastatic ability compared to the parental cells, as you can see in the bottom, where lung metastasis by Telvan injection were obtained for the parental cell line, some 159, and MCF DCIS. And the metastatic mass was higher for injection of hybrids between these two cells, these two cell lines. So that's interesting in terms of phenotype. What we were interested in as well is how the phenotype is passed on to progenies, and what kind of genetic rearrangement occurs after fusion. And so the main hypothesis was that once the two parental cells fuse, then asymmetric divisions occur. And so some cell get a lower ploidy than the hybrid parents, and some cell get a higher ploidy. And so the cells with lower ploidy are selected for because the ones with too much genetic material can't make it. And so you have progressive recombination with ploidy reduction with generations. So in order to observe that, whether that is the case or not, a ploidy analysis was performed for the hybrids. And so in these plots, you see distribution that are bimodal. And so the first peak corresponds to the cells that are in G1 phase, and the second peak corresponds to the cells that are in G2 phase. And so this is basically the DNA content. So the two, white distribution are the parental cells, and the blue distribution is a hybrid between the two. And so P4 corresponds to passage 4. So basically the hybrids were obtained, and then passage 4 times, in culture. And you see that at passage 8, so after they sustain a number of doubling, you see that the peak shift to the left, which means a reduction of the DNA content, and so ploidy reduction. And that was observed for a certain number of replicates. So that's about for the ploidy reduction. But what was very interesting as well to investigate was the chromosomal content. And to investigate this, different parental cell lines were used. So typically MDA, MB231, and SUM159 that are, patient-derived cell lines for which the SNPs are known. So the SNPs are single nucleotide polymorphism, and that those are part of the DNAs that are different between the patients. And so all the SNP array is known for these 2 cell lines. And basically, if we obtain a hybrid from those cell lines and perform an SNP analysis, we can tell what part of the genome is from which parent, which is very cool. So, for instance, on the D, so the x-axis corresponds to going along the chromosome, and here are plotted chromosome 13, 1, 20, and 21. And the green parts are the ones that contain like SNPs from 2 parents, which means a heterozygous, part. Whereas the yellow is just SUM159 and the blue is just MDA. And so those yellow or blue part correspond to loss of heterozygosity. What you can see is that there is some variation, between the hybrids, because B2, B1, A11 are different replicates, different hybrids from, the same like fusion between the parental cell lines. And so it means different kinds of recombination across the different hybrids. So at left, you see a similar thing for 2 other cell lines.
Is it just me or?
Yeah, no, I think that we lost Etienne.
Oh.
Yeah, because yeah, he seems to be very focused on that slide, but
Is it just me or?
Well, because I'm thinking like, is my internet?
Oh, he's gone now. Yeah.
it's gone now. I know he's back. He's back.
Hi. So, okay, I'm sorry. I just realized probably my connection froze. I was speaking alone, probably for a certain amount of time.
David does that whenever somebody talks for too long, David switches up the connection.
I see. Okay, then maybe I'll conclude. So I don't know actually what did you guys miss? Did you get that part, or did you miss it as well?
No, this one we didn't.
You didn't get it. Okay. Can you hear me?
Through it. Yeah. So you went through the next one as well.
Oh, I went through it. Okay.
Yeah. Yeah, this one that you were stuck on.
Okay. Yeah. So the left plot correspond to basically the same thing for two other cell lines. And those plots are already very informative, but what they don't tell is about the copy numbers, because you could interpret easily those, chromosomal plots if the cell lines were to be deployed, right? But because it's not the case, it's interesting as well to look to the copy numbers. And what you can see, for instance, at the right is that basically the MDA cell line has a lot more than two copy numbers for most of the chromosome, whereas the HS578 cell line is mostly deployed, and you can see that the copy number variation amongst the hybrids resulting from fusion is even more diverse amongst the hybrid, because, for instance, the hybrid A2 has very different copy number than the two others. So you see, like, really diversification not only in terms of DNA, like nucleotides, but as well in terms of copy number. So this shown the impact of fusion on genetic diversification, but maybe even more importantly, what's the impact on phenotypic diversification? And for that, Megan and Philip analyzed some single RNA-seq data from those hybrid cultures, and characterize some clusters for the hybrids versus the parental cell lines. So in the B plot, you see the UMAP axis, which are kind of similar methods as TSNE, and it shows, like, that the MDA parental cell line cluster form one cluster, and the SUM159 form a cluster as well, but the hybrids between the two, after just two passages, are very close to one of the parental, but after some passages form the sort of own phenotype. And so, interestingly as well, from this single cell RNA-seq, Megan could quantify those diversity index in the population. And what we see is that after just passage 2, which is, pretty much right after fusion, right? The diversity is very high, the diversity of the population. So we see this really diversification mechanism in action. But after a certain number of passages, probably by selection and sort of selection against the misfits, the amount of diversity comes back to very close level to the parental cell lines. So what we're interested as well is the impact of these potential genetic recombinations after fusion on cell population diversity. And for that, mathematical modeling can be a useful tool, because we can basically model an asexual versus parasexual process, in the sense that we can run a simulation with mutations only and compare the amount of diversification when now fusion is accounted for. So the way we model that is a Poisson process that will help to model a branching process in which we start with one cell with a wild type, and so represent the genome with an array of zeros for the wild type and 1 when a mutation is present. The cell division, so the number of newborn per time iteration, is modeled by a Poisson process. We have as well a density-dependent death term. And here we don't consider any impact of the mutation on the fitness for the moment. So sort of a neutral evolution case. The way we model fusion is we have, every time step a probability of cells to fuse with another cell of the population. And when that's happening, we consider a random recombination between the mutations of each locus. And so as well, the reason why we don't consider any impact on the fitness at the moment is that the goal of this simulation is more to show the role of fusion in diversification on the population level, and not necessarily in terms of further selection. So really to see the sole effect on diversification, neutral evolution is probably the most simple and suited case. So in order to, so here we have a fusion probability that is one of the parameters of our model. In order to, have a sensible value for this parameter, realistic value, what we did is we considered a simple ODS system of exponential growth that mimic the in vitro co-culture between green and red cells. And given the final number of double positive hybrids, we use that number to estimate the fusion rate. And then so obviously that doesn't estimate one fusion rate, but a range of fusion rates, because a lot of different replicates and cell lines were used. And when we look to realistic mutation and fusion rates, what we see is that when comparing simulation with mutation only to mutation and fusion, we see a dramatic impact on diversity, especially on clonal richness, which is just the number of total mutants present in the population. So that's the first plot, and the other plots are different metrics of diversity. So the Shannon index, the Simpson index, that accounts as well for the frequency of mutants for each genotype. But you can see overall that you see a significant, difference between with or without fusion in, a clinically sensible time. Like here, it's a simulation of 3 years. And these are two examples of most plots with the two cases where what you see on terms of clonal richness reflects on the number of mutants you see in this most plot. And we perform that for a large range of parameters for mutation and fusion rates in considering realistic range. So for fusion, basically the rates we estimated from the experiments, And for mutation rates range from the literature. What you can see is that for really low level of mutation rates, you don't see a lot of diversification brought by fusion, essentially because for low mutation rates, the population is very low in terms of heterogeneity to start with. And so when fusion happens, usually it's between cells that are identical. So fusion doesn't have any diversification potential in that case, but you can see some gradients when the mutation rates get higher. And those are the richness plots for the different parameter values. Interestingly as well is the impact of fusion on a pre-existing heterogeneity in terms of diversification. So here what we plot is the final heterogeneity, clonal richness after a simulation of 3 years. And the x-axis is the pre-existing heterogeneity in the population. So, for instance, so it started at 1 cell until and go to 50 pre-existing mutants, and you see that the fusion mechanism will really power up the sort of diversification from pre-existing heterogeneity. And this was observed for several parameter sets in the ranges that we consider. So this is very cool because it shows that fusion can have an impact on population diversity, even for realistic fusion rates. Even if this mechanism, those events are fairly rare, it has an impact potentially on the overall diversity. However, in the previous non-spatial simulation, we consider fusion between any cell of the population, which means that basically the population is well mixed, and so a cell is susceptible to fuse with anyone. But we know that in reality, in solid tumors, that is not the case, and cell will be able to fuse only with its neighbors, and its neighbors have a good chance to be actually fairly similar, genetically, because if you have sort of these subclonal clusters that look alike, that's really likely. So potentially the impact of fusion on considering the space is actually lower than we thought. So in order to account for this, we develop an agent-based model that will reflect basically the same process as the non-spatial model, but in a special setting. So first, what we had to do is estimate the spatial fusion rates, basically. So was the probability, per basically unit of time for a cell to fuse with one of its neighbor. And to do that, I ran a simulation that sort of mimicked this green-red co-culture, and at the end counts the number of yellow hybrids, try to calibrate the fusion probability to match this number of double positive cells. And so that's an example of this kind of simulation. So it's a sort of 3 days co-culture, and you see those big yellow cells forming. And then from those estimated fusion probabilities, now we run the sort of spatialized diversity model where we start with 1 cell, and we basically look to the final like clonal richness or any diversity metrics. And so yeah, so this is the two simulations. I mean, just an example because I did several runs of them just to show how basically the model looks like. So at left, that was with mutation only, and at right with mutation and fusion. And we did the same in 3D. And we did in 3D because basically in 3D, a cell has more neighbors to fuse with. So potentially they could make a difference. And so over the course of the simulation, over a year of simulation, we see already a statistically significant difference between no fusion and fusion, and even more difference when you consider fusion 3D. So yeah, the conclusions of this study is that cell fusion is a source of genetic and phenotypic diversification. These are rare events, but some cells are found to be, some cell cultures are found with 1% of hybrids. So it's not that rare. Hybrids tend to be more migratory and metastatic. Computational simulation predict that fusion enhances population diversity. And then, I guess there are still very interesting question that remains. And one of them is, what's the impact of a treatment, efficacy or resistance? So what's the impact of fusion hybrids on these? Can potentially, fusion brings enough, diversity in a population to lead to a heterogeneity that will favor presence of resistant clones. Another question that is maybe less clinical and more theoretical would be, for small cancer population, can fusion be a way to avoid like population decline? In passing, in conserving maybe good copies of all good mutations and getting rid of deleterious mutations. Okay, I guess I'll stop now because I think it's 1 hour and a half already. So I was pretty much at the end anyway, but so yeah, with that, I would like to thank, well, of course, David, that mentored me for all these 5 years, and that was really a pleasure to work under his supervision. And Connor was, very helpful in the macrophage project, as well as how. I would like as well to thank everyone in the IMO to be such a group of pleasant folks, and I'm a bit sad to leave. I think the last year was a bit weird because we couldn't really catch up as much as we usually would do, but it was really, I felt really like in a big family here. And I would like as well to acknowledge everyone that you know, contributed to the fusion projects. That was a very cool project. We got a cool publication out of here in Nature, Ecology and Evolution, and that's my next step. So I'm gonna leave, maybe temporary, who knows, but Math Onco, and go more into multicellular evolution, and follow you know, another patient of mine with potentially field trips to look to fossils. So I'm gonna be with Emma Lund in Lund University, investigating the impact of oxygen increase on invertebrate evolution during the Devonian. So, but you know, I'm still interested in pursuing some of the projects let in suspend in the cancer field. So I'm still reachable, and I'm glad to collaborate. So don't hesitate to contact me. So yeah, thank you.
Thanks, Etienne. Quite a tour de force, and I'm sad to see you doing different research, which looks very, very exciting. I'm still not giving up the hope that at some stage you'll be coming back to Math Onco. I have your contact details, but other people I might want to reach you later on. I don't think you're leaving no Slack anytime soon, but clearly email is always a good way to reach you, I guess. But
yeah, I'll still be on Slack too.
That's right. It's definitely way past 1:30. If people need to ask questions now, that's great. Anyone else who wants to stick, please stay. Definitely other people might have other things to do, so you want to leave, this is a good time too. And thank you once again, Etienne. This has been incredible.
Yep.
Thank you guys for your attention.
I just want to reiterate that it's been a pleasure working with you over the years, and thanks for all your input into HAL and really being part of the IMO team. It's nice to hear you say that you were part of a big family, because that's really the way I view IMO, and you definitely were one of our sons. So good luck with your next career steps. I'm sure you'll be successful whatever you do. But please come back and work in Math Onco in the future.
Thanks, Sandy. I really appreciate it. And yeah, that was a pleasure for me too, again.
Okay, thank you. So I've got to go, unfortunately. So I'll say goodbye.
Cool.
Bye, Sandy.
Bye, Sandy.
Great talk. Thanks.
Thanks, Philip.
Cool. All right then. Thanks again, everyone, for coming. And some of us are just going to go to World Beer now to have a couple of drinks and maybe some food. I'm hungry now. You're welcome to come and join us as well.
Is Etienne going to drive from home?
I hope that he's going to. I mean, it's a bit around, but it's still possible. He definitely is expected to come, yes.
Okay. Suspected or expected?
To World of Beer?
Expected, yes.
Oh yeah, I'm going, definitely.
Okay, so I'll see you there in, I don't know, half an hour, or when? How long would it take you to get there?
Half an hour is probably a good time, yeah.
Okay, cool. We'll see you.
Sounds good.
Bye.
Bye.